Hello to everyone watching this video. My name is Christinendinya and I'm going to present a project on the effect of chicken manure on leaf fields of selected African leafy vegetables in Western Kenya. This work was done by staff from Cleome, Anglican Development Services and Agro-Kenya, all found in Western Kenya. African leafy vegetables are important for the health they provide for food security and income. These vegetables require sufficient amount of fertilizers to give good yields. Unfortunately, artificial fertilizers are not so easily affordable for our small-scale farmers, but chicken manure can be an alternative source since most of them rear chicken in their farms. But the suitability of chicken manure produced by these farmers and the effect of chicken manure in the vegetables is not quite known. So in the study, our objectives were to investigate the presence of the phytopathogens in the manure, which would eventually affect the yield of the crops during growth, to determine the nutrient contents in manure samples, and to demonstrate the effect of the chicken manure on leaf fields of selected African leafy vegetables in Western Kenya to our farmers. The study was done in Busia, Kakamega and Vihiga counties in 2018, and 15 farmer groups participated. We collected 1 to 2 kilograms of local chicken manure samples from these participating farmers. These samples were tested for parasitic nematodes, pathogenic fungi, and bacteria, and the nutrients at our cultural laboratories in Nairobi. To demonstrate chicken manure performance, we set on-farm trials where we planted spiderplant, Amaranth, African nightshade, cowpea, and slenderleaf. And these were done in 2 sets. One set had 10 metric tons per hectare of manure applied, and the second had 0 manure of chicken manure applied. The experimental design was randomized complete block, replicated 3 times. Plot sizes were 9 meters squared. Data was collected on plant heights, fresh and dry weights of yields at 15 days after planting. Results showed that there were no parasitic nematodes nor pathogenic bacteria. However, 44 of the samples had the pathogenic Fusarium, which is a fungus. The nutrient analysis, which is summarized in table 1, shows that the samples from Vihiga had the highest average nutrient contents for the nutrients shown. Kakamega was high just for phosphorus alone, and Busia for calcium and copper. The leaf yields from the plots with chicken manure were significantly higher than those without chicken manure in spiderplant, amaranth, and nightshade, but this was not the case in slenderleaf and cowpea. So the absence of parasitic nematodes and the bacteria was a good thing because the chicken manure from the farmers would not infect the crops during growth. The nutrient content in chicken manure was sufficient to increase the leaf yields of the 3 vegetables, nightshade, amaranth, and spiderplant. The leaf yields realized for nightshade especially were high compared to the reports that show that 1.3 to 1.5 metric tons per hectare is the average yield from the farmer. Lack of difference in leaf yields of slenderleaf and cowpea when planted with and without manure could have been because of the nitrogen fixing ability of the 2 species. We can conclude that chicken manure contributed to the increase of the leaf yields of nitrogen, amaranth, and spiderplant, but not cowpea and slenderleaf. I would like to acknowledge the National Research Fund for funding this project, the cultural director for his support, even as we carried out these activities, and the farmer groups in the mentioned counties for participating in the project. Thank you very much.